Hello, Saravanan.
Sir.
Sir is coming.
Good morning, sir. Good morning, sir. How are you, sir?
Morning.
Fine, sir. Fine, sir. Fine, sir.
So
Thank you,
in
sir.
college or?
Yes, sir. I'm in college only, sir.
Not very clear.
கேட்கலையா? சார் கொஞ்சம் மியூசிக் குறைங்க. Yeah. Yes, sir. It's audible now.
Sir, good morning, sir.
உங்களுக்கு தான் கேக்குதா?
Sir, am I audible, sir?
Hello.
Hello.
Sir, am I audible?
எனக்கு கொஞ்சம் இந்த not very clear.
Sir, good morning, sir. Sir, am I audible?
You are audible, ma.
Okay.
Yeah. Saravanan?
Sir. Okay, sir.
Good morning.
Good morning, sir. Good morning, sir.
Sorry, it is not audible.
Okay. How are you, sir?
Very good. Making this type of seminar and interaction with the people.
Yes, sir.
You are staying in Hyderabad, coming to home?
Yes, sir. I was in Hyderabad only, sir. I was stuck in Hyderabad. Last 4 months I was in Hyderabad.
So another 6-7 months also you have to be there.
So it is very difficult, sir, because family people, sir, they are missing very much. I am also missing my family, sir.
Oh, family is not with you?
Yes, sir. They are in Tamilnadu, sir.
So they are safety?
Yes, sir. Yes.
Okay. Good.
Yes, thank you, sir.
Thank you. Participant.
Okay, shall we start the session? Is it audible?
It's audio.
Okay. A pleasant morning to all. Thank you for joining us for today's webinar. I am Mrs. Kirtika, Associate Professor, Department of Pharmaceutical Chemistry, Nandha College of Pharmacy, Erode. Today we are presenting a national webinar on the topic of nanochemistry and overview, present by our resource person, Dr.G.SARAVANAN, Head and Professor, Department of Pharmaceutical Chemistry, MNR College of Pharmacy, Telangana. On behalf of our institution, I feel grateful to invite our participants from various institutions on this fruitful virtual platform. I assure that there is no need of introduction of our resource person who have an intellectual personality. So today's webinar is quite interesting with the topic nanochemistry. It's a combination of chemistry and nanoscience. Nanochemistry is associated with synthesis of building blocks, which are dependent on size, surface, shape, and defect properties. Also, it's quite useful in chemical materials and physical science, as well as engineering, biological, and medical applications in a great way. We feel proud and happy, Saravanan, by connected with this virtual platform today. We would like to express our gratitude to our chairman, Thiru B. Shanmugan, and to our secretaries, Thiru S. Nandakumar Pradeep, Nanda Educational Trust, and Thiru S. Tirumurthy, Nanda Educational Institutions. Without their motivation and support, organizing this webinar is highly impossible. Let us now welcome the man who doesn't need any introduction and known among all of us for his wisdom and his knowledge. Yes, it's our honorable and beloved principal, Dr. T. Shivakumar, Nanda College of Pharmacy. I would like to request our principal, sir, to deliver the welcome address, please, sir.
Okay, thank you, Kritika, for the introduction
Thank
of
you, sir.
this warm good morning to one and all. I heartily welcome you all on behalf of Sri Nanda Educational Trust for this prestigious occasion of our institution for national webinar on the topic of nanochemistry and overview. I feel immense pleasure for the august presence of our eminent speaker, Dr. G. Saravanan, Head and Professor, Department of Pharmaceutical Chemistry, MNR College of Pharmacy, Telangana. I congratulate Saravanan, you are the maximum participant, almost 1,455 registered, and Kritika staffed for this further registration. In this moment, I extend thanks to Kritika to bring to our webinar session. Thank you, Kritika,
Thank you,
for
sir.
this It is my pleasure to welcome you for this platform, Saravanan. Welcome.
Yes, thank you, sir.
Yeah, I'm happy to have more than 1,455 participants from various pharmacy colleges and universities in and around India, in which one of the international delegate, Anu Rosina, King Hamad University Hospital, Bahrain. And I feel proud to invite some delegate from other part of India, Dr. Kamal Kishore, MJP, Rohil Gandhi University, Bareilly, Uttar Pradesh, Dr. C. Sadak Valley, Avanti Institute of Pharmaceutical Science, Hyderabad, Professor Amit Sharma, Jagannath University, Jaipur, Rajasthan, Dr. Biswajit Das, Himalayan Pharmaceutical Institute, Gangtok, Sikkim, and Dr. Dev Prakash, Dagania Accurate College of Pharmacy, Noida, Uttar Pradesh, and Mr. Assis Singh, Lee Ford Healthcare Limited, Paddy, and other delegates from Andhra Pradesh, Kerala, Telangana, Karnataka, and Himachal Pradesh, and many of the participants from Tamil Nadu, Uma Maheshwari Madam from Naiga, Intulada from the Coimbatore, and other many friends from the long associated with this webinar. Thank you all of them. This webinar highlight about nanochemistry. It is an emerging new field of chemistry, in particular of solid state chemistry, which emphasizes the study of development of preparation method of useful material with nanometer size dimension. Nanotechnology in medicine currently being developed involves employing nanoparticle to drug delivery, heat, light, or other material or substance to the targeted cells. This topic will be innovative in the view of chemistry aspect. Definitely, it could be a reach to our delegates by our resource person, and he is very good interpretation data in mass spectra, NMR spectra, IR spectra also. He has various field in the different level. I feel it should be a fruitful one to all delegates. I feel greatly indebted to our chairman, through V. Sanmugan, and to our young and energetic personality, through Nandakumar Pradeep sir, Secretary, Sri Nanda Educational Trust, equally to through S. Tirumurthy, who is innovative and incorporated to our institution, Secretary, Nanda Educational Institution, Erode. Their constant encouragement towards this event has opened new avenue for further build the institution in terms of its reputation and quality. Also, I feel grateful to our Chief Administrative Officer, Dr. Krishnamurthy, for his moral support to this function. And equally, I extend my thanks to the team and Mrs. Kalpana, Gladys, and Dr. Jagdishwaran, and equally Hemalata from the pharmacy practice department, and from the chemistry department, who is always running behind this seminar, mobilizing all level and torturing me also, is Mrs. Kritika, and young and energetic administrative officer, medical college, Ayurveda, Siddha, and other associate work, Professor Apollo James, for this moral support of this event. I extend my gratitude to faculties for their effort taken and hard work put forth for this function in a successful way. Last but not least, once again, I am welcome to our dear most participant with the colorful faces from various institutions for this function, and the student of Nanda College of Pharmacy and other College of Pharmacy in and around India. heartful wishes and keep on updating your happily. Thank you once and all. Thank you.
Thank you, sir, for your kind and valuable address to all. Thanks a lot, sir. It's an important announcement for our delegates. So, as usual, kindly follow the session, and you are invited to discuss your valuable suggestion and your doubts regarding this topic. Should we enter into the question and answer box or chat box on your control panel? It will be discussed with our resource person in questionnaire session. So please join with us with your cooperation and appreciation. It should be a successful event today. And now we invite Mr. Apollo James, Professor, Department of Pharmacy Practice, Nandha College of Pharmacy, to introduce our resource person. Please, sir.
Good morning, everyone. On behalf of Nandha College of Pharmacy, I'm very happy to introduce our today's resource person, Dr.G. Saravanan, HOD and Professor, Department of Pharmaceutical Chemistry, MNR College of Pharmacy, Sangareddy, Telangana, India. Dr. G. Saravanan received his master's from the Tamilnadu Dr. MGR Medical University, Chennai, in the year 2007 with distinction, and received his doctorate from Jawaharlal Nehru Technological University, Hyderabad, in the year 2014. He is having more than 14 years of academic experience. Currently, he is working as professor and head Department of Pharmaceutical Chemistry, MNR College of Pharmacy, Sangareddy, Telangana, India. He has received the Best Achiever Award in 2019 and Best Teacher Award in 2018 and Best Researcher Award again in 2018. Two of his research findings were patent filed. So far, he published 72 articles in reputed national and international journals, and 89 papers were presented in various conferences. He also published a book, and he is a chairperson for more than 10 conferences. He is a PhD guide in Osmania University. So far, he guided more than 31 MPharm and 10 BPharm projects. He delivered various guest lectures in various institutions and several GPAT academics. He is a reviewer for many national and international journals and editorial board member of Anti-Infective Agents, Bentham Science Publisher. His areas of interest were in novel drug delivery and study of its therapeutic importance, analytical and bioanalytical research, design and synthesis of small and macromolecules involving multi-step reactions. He has wide experience in the development of new organic methodologies, competent in handling organic reactions in both multimode and monomode microwave reactor, and purification of organic compounds by column chromatography. He has experience in interpreting analytical and spectroscopic techniques such as IA, NMR, mass, and microanalysis. Hands-on experience in NMR predictor, mass fragmenter, and associated softwares. With this brief introduction, I am handing over the session to Dr. Saravanan. The screen is yours.
Thank you, sir, for your nice introductions. So, a very good morning to one and all. The first thing I would like to thank that management of Nandha College of Pharmacy, particularly through V. Sanmugam, Chairman, Nandha Educational Trust, Mr. S. Nandakumar Pradeep, Secretary, Nandha Educational Trust, and Mr. S. Tirumurthy, Secretary, Nandha Educational Institutions, and the convener of this today webinar, Dr. T. Sivakumar sir, and other organizers, Dr. M. Jagadishwaran, Mrs. N. Kritika, Mr. Apollo James, Mrs. Gladys Kalpana, and Mrs. S. Kamalada for giving this a big platform to discuss about my view on nanochemistry and over. I wholeheartedly welcome all the delegates for this webinar on nanochemistry and overview. So in the today discussions, we are going to know about what is a nanoscience and what is a nanotechnology and what is a nanochemistry and what indicates that nanoscale. And we are going to discuss most about that nanomaterials, which was the materials behind that nanochemistry. What are all the different types of your nanomaterials and how these nanomaterials are characterized and what are all the important properties of these nanomaterials and how these nanomaterials are compared with that bulk materials and how these nanomaterials are synthesized and what are all the disadvantages which are associated with these nanomaterials and what are all the major applications of these nanomaterials. And I'm going to explain this nanochemistry with one case studies which what we have done in our laboratories, and finally from that particular nanochemistry what we are concluded and the references that we will discuss one by one. First thing, nanoscience. So always when if you want to understand about any term, when it was on a complex, it is difficult to understand. When we are splitted the complex terms into on a simple parts, it is very easy to understand. So in that nanoscience, that we are splitting the terms into one or two things, a nano and a science. That nano indicates nanomaterials and the science. I think we people may aware of that science. Science is nothing but it is a knowledge of your systems, or we can tell the science is a knowledge addressing the general truth. So a nanoscience that will discuss about the knowledge of that nanomaterials, which are about including a theory and experiments which will related to the properties and phenomena which are associated with that nanomaterials. So simply if you want to tell means it is nothing but study about the structures and materials which are on that ultra small scale. So what will happen when that materials are in that ultra small scale? At this ultra small scale level, the physical and the chemical properties of the matter will change. So that is the thing why we are talking about these nanomaterials. And this nanoscience is not restricted to on any single field, and it was combining on a many cross disciplinaries. So these nanoscience is studied by chemistry peoples, physics, even biologists, medicines, computing and material science people are studying, and even engineering are studying about that nanoscience. Why means to understand better about that nano world. So this nanoscience is not restricted for any disciplinaries. There are many disciplinaries they are studying about this nanoscience. And coming to that nanotechnology. Again, here also we are splitting the term into one or two parts as a nano and technology. That nano indicates a nanomaterials and technology. What is technology? Technology is nothing but a practical application of the knowledge. So, which includes the development, application, and commercial implementation of that knowledge what we are gathering. So, nanotechnology is otherwise known as a molecular manufacturing. So technology here, we are applying the knowledge of the system to produce a design, production, and application of the any structures, devices, and systems at the nanoscale. So if we produce or prepare any structure or devices or systems which are at this nanoscale level, so that particular technology we will call it as a nanotechnology. So if you, these nanotechnology and a nanoscience, these both are interrelated. So simply nanoscience is studying about that nanomaterials and its properties. Whereas if you are applying those nanosciences for preparing on something new or different products or devices, so that we can call it as a nanotechnology. And coming to that our topics, nanochemistry. Again, it is a combination of a nanoscience and a chemistry. And we people may know about chemistry. It is nothing about study about the chemical reactions or chemical compounds. And just now we discussed about a nanoscience. It is nothing but a knowledge of the system which includes a theory and experimental part which discuss about the properties and phenomena which are associated with that nanomaterials. Okay, so this is the combination of the chemistry and a nanoscience. So simply if you want to tell means this nanochemistry is associated with the synthesis of your building blocks. So these synthesis of the building blocks will be dependent on the size, surface, safe, and defect properties. And this nanochemistry will be very useful in the chemical and material sciences, physical sciences, as well as in the engineering, biological, and medical applications. And in all these departments, that the concepts will be same, but how that was used, that particular concept. So the usage of the particular concept will be different in this field, but the core concept in all these disciplines are the similar only. So till now we discussed what is nanoscience, nanotechnology, and nanochemistry. In all these cases, we had the common term is nano. So we should understand about what is that nano. So in the Greek, that nano indicates means dwarf. So that means it is on a small. And in the dimensional, or if you want to discuss about that nanoscale, we will denote by means of a nanometer, and that nanometer is indicated by nm. And the nanoscale is usually between that 1 to 100 nanometer. Any dimensional which was between that 1 to 100 nanometer, we can call that one as a nanoscale. So if we take an a meter, that 1000 meters, we will call it as a kilometer. So this is a simple thing that every people we may aware of that one. And 1,000 of that meter, we will call it as a millimeter. In that millimeter, against a 1 in thousands, that we will call it as a micrometer. And this micrometer range, we will call it as a microvalve, and usually it cells, bacteria, and silicon chips, all those things will come in this microvalve. And in that micrometer, again, yeah, 1,000, we will call it as a nanometer. In the meter, if we want to tell, it is a 1 billionth of a meter only, we will call it as a nanometer. So that is nothing but a 10 to the power of minus 9 meters. So that is nanometer. So that a dimensional range of 1 to 100 nanometer. So that we will call it as a nanoscale. So for an easy things, how big this nano, for that I was giving this structural explanations. So people may aware of that atoms and molecules. So the hydrogen atoms, if you are placing a 10 hydrogen atoms side by side, so the total length will only a 7 nanometer length. So we people may aware of that nucleic acids. In that, the genetic information is carried by a DNA, and it is on a double helical structure, and the width of that double helical structure of a DNA is only 2.5 nanometer wide. And we may aware of that red blood cells. So the wideness, thickness of that red blood cell is 7,000 nanometer. And day to day we are using a paper. So the thickness of the paper, if you are looking, it is on a 75,000 nanometer thick. Then you think how small this nanometer. And we are pinhead having on a 1 million nanometer wide, and our human hair is 50,000 to 1,000 nanometer wide. So that much small this nanometer. The size of the ant is on a 5 nanometers, and the dust mite is 200 micrometers. In that human hair, I told it is on in the nanometer hands. I told here it is on a 10 to 50 micrometer. The red blood cell is having 2 to 5 micrometer wide, and DNA is having 2 to 12 nanometer. And if we are coming to micro electrochemical devices, the size is 10 to 100 micrometers. And in the quantum coral, in that the iron atoms, 45 iron atoms, the size is only on a 40 nanometer diameters only. And a carbon nanotubes, which is having on a 2 nanometer diameters. And this is on a nanometer scale, as I told, 1 to 100. So this is called as a nanoscale. Anything which is coming under this 1 nanometer to 1,000 nanometer, that one only we will call it as a nanoscale. And people may aware of that a tennis ball. So tennis ball is how much compared to that nanometer? It is on a 10 to the power of 8 nanometers. The salt grain is having 10 to the power of 6 nanometers. And a cell is having 10 to the power of 5 to 10 to the power of 4 nanometers. And the size of your virus is 100 nanometers, and DNA, as we told, that is on a 2 to 12 nanometers, and the structure of your proteins, the size is between 1 to 10 nanometers, and the glucose is 1 to 0.1 nanometers. And polymers will come under the category of your 10 to the power of 6 nanometers, and quantum dot will come between your 10 to the power of 4 to 10 to the power of 5 nanometer, and your gold nanocell will come under that 1000 nanometers, and dendrimers having the size of 10 to 100 nanometers, liposomes having a size of 1 to 10 nanometers, and the micelle is having a size of a 0.1 nanometers. So again, for an easy understanding, and now I'm going to play a video of that nano valve. What is nano? Nano is a unit for measuring extremely small size measured in the nanometers, which is the 1 billionth of a meter. How much is the billionth? For example, if you look at emperor penguin, which is having 1 meter long, if you are zooming the emperor's penguin skins, the tick is having 1 in 1000 meters. The bacterium attached to the tick is about 1 billionth of the meters. So the bacterium's DNA, so the size of the DNA is 1 billionth of the meters. So 1 nanometers. So DNA is made up of many atoms, and atoms are made up of all matters. That is called as nano valve. And this nearly invisible valve can be viewed with an atomic force microscope. So now you people may know how big these nanoparticles are, nanomaterials. So in all this case, nanochemistry, nanoscience, nanotechnology, in all this case, we are discussing about on a nanomaterials. What is these nanomaterials? These are materials which changes its properties at the nanoscale level. So any material, if its properties are changing at this nanoscale, that nanoscale means already we know it is on a 1 to 100 nanometer. In that particular size, if any material is capable to change its properties, that materials we can call it as a nanomaterials. So what are all the properties may change? It may be on a conductivity, color, and mechanical hardness, all those things. So that properties is changing at the nanoscale, we can call that materials as a nanomaterials. So nanomaterials will encompass all nanoscale materials or materials that contain at least 1 nanoscale structure. So that means that materials must have a 1 nanoscale structures that may be present either in their surfaces or it may be present internally. So these nanomaterials may be an inorganic in nature or organic, or it may be on a biological nanomaterials. So these are some of the examples of that nanomaterials. That organic nanoparticles, if you want these things, dendrimers, layer biopolymers, liposomes. So that will come under the category of your organic nanoparticles. And in case of your inorganics, that carbon nanotubes, mesoporous silica, gold cells, iron oxides, quantum dot, all those things will coming under that inorganic nanoparticles. And the hydrogen will come under the category of your hybrid nanoparticles. And nanomaterials, what are all the different types of the nanomaterials? The nanomaterials are nanoplates, nanoparticles, nanowires, and nanotubes. So these all things are which was prepared or synthesized in the lab. So that's why we will call these nanomaterials as engineered nanomaterials or lab nanomaterials. And some of the nanomaterials which were available in the nature also. A very good example is smoke, sea spray, volcanic ash. All these things which was occurring are available in the nature. So we will call these one as a natural nanomaterials. So these nanoparticles, nanoplates, nanowires, and nanotubes, all those things are distinguished by means of their shapes as well as in the dimension only. So the difference between all these nanoplates, nanoparticles, nanowires, and nanotubes are only thing is shape and the dimension only it was different. And the common thing behind in this nanomaterial is that one or more dimension of these materials should be in the nanoscale. So which thing we will call it as a nanoparticle? All the three dimensions, that means length, width and height. All these three dimensions are present in the nanomaterial. And nanomaterials are
Hello.
hello.
Sorry, ma, some network issues, I think so. Is it audible
Okay.
now?
Yes, it is
Yeah,
audible.
okay, okay. Sorry for the interruption.
So that minimum one dimensional should be in the nanoscale. So in case of a nanowire, the diameter is the nanoscale, but the length is not in the nanoscale. Whereas in case of a nanoplates, if we take and the thickness is at the nanoscale. And in case all these three dimensional, that is on a length, height and width, then we can call that one as a nanoparticles. And here that nanomaterials, that already I told that nano means in the Greek it is dwarf or little old man, we will tell. So that will be differ in their size, surface, shape, and that we will have on some nanomaterials here. The size, as I already told, it is on a between 1 to 100 nanometer. That scale, if it is that, we will call it as a nanomaterials. And in the surface, it may be on a PGlations or other coatings, or the functional groups may be present, or the surface charge may be responsible for that nanomaterials, or it may that targeting the ligands. And that shape may be on a sphere shape, or it may be on a cube shape, or rod shape, or star shape, or it may be like on a plates. And the materials examples, it may be on a protein drug conjugates, dendrimers, carbon nanotubes, metal properties, polymer particles, hydrogen particles, and solid lipid hybrid particles. How we can characterize these nanomaterials? And the characteristics which are responsible for the properties of a nanomaterials are structure, particle size, and surface. So these three things are mainly responsible for the property of a nanomaterials. And these structure, particle size, and surface characteristic can be studied by various techniques. And these are all the some of the important technique I was listed out. That X-ray diffraction technique and low energy electron diffraction, simply we will call it as LEED, and SEM, that is scanning electron microscopic, and AFM, that is nothing but atomic force microscopy. All these instrument techniques are the major techniques which are used to characterize these nanomaterials or nanoparticles. And what are all the properties which are associated with these nanomaterials? So when that compounds are going to on a small, ultra small, as the physical and chemical properties are changing. So that is on a conditions for nanomaterials. So when it is break down into a smaller particle, ultra smaller, that conditions, if it is changes its physical and chemical properties, then we can call that one as a nanomaterials. So a simple example I will tell, that's a lump of your gold. I think we people all may know about on a gold, and the color you people may tell immediately that the color of that gold is on a yellow, as in the bulk properties. When that gold was
broken or break into small pieces in the range of 10 to 100 nanometer, the color will be changes into a reddish. Sometimes it may be an orange, purple or green, and these colors are depends upon the size, shape of that gold particles. At this size level, at this minute level, that gold can act as a catalyst, and it will be very vigorously active or increases the rate of the particular reactions. So the reason behind of these changes, that means there is no change in the chemical composition is taking place. Only thing that a more surface area is exposed. Because of that more surface area exposed only, the color will be changed from that yellow to a reddish, orange or a purple. So then you think about the things, how these nanomaterial properties will be useful. And as we know that properties are changing due to that nano-sized particles, and this particular thing we are keeping in the mind, and we can able to control the various fundamental properties of that nanoparticles or nanoparticles, and we can able to build many molecules. So by means of keeping a color, electrical conductivity, melting temperature, hardness, crack resistance and strength, all these things we can in a controlled manner we can keep, and from that we can able to build a molecules, and that can be very useful. And when that particle size are decreased, ultimately what will happen means the surface area to a volume ratio will increase dramatically. Because of that increased surface area only, that the properties are changing. And coming to the chemical reactions, so generally the chemical reactions, the catalyst actions will take place at the surface level only. And when the particle size are small, ultimately the reactions will be on a very vigorous. And more surface area, ultimately what will happen, that it may leads to a more catalytic actions. If that catalytic action is more, the reaction speed will increase, and if that reaction speed is increased, ultimately we may able to save that energy. So that is the advantages of this nanochemistry. So when we make these things as a smaller size, the catalytic activity is increasing. Because of that increased catalytic activity, the speed of that particular reaction is increased. If the speed is increased, ultimately we can able to get an energy efficiency. And sometimes the quantum properties will also come under this nanoscale. That's why sometimes we will call this nanoparticle as a quantum dots also. And I'm not going to discuss about all those properties of that nanomaterial. Some important 4 properties I was chosen, and I'm going to discuss elaborately about these 4 properties only: mechanical properties, catalytic properties, magnetic properties, and optical properties of your nanomaterials. Mechanical properties. So if we take a long hollow cylinders, so generally we will call that one as a carbon nanotubes. So this is called as a carbon nanotubes. How these carbon nanotubes are formed means when we are rolling up this graphene sheet, we can be able to get these carbon nanotubes, and this carbon nanotube is one of the strongest material which was available in the nature. And if you are looking the geometry of these carbon nanotubes, so it may have a different properties in the radial direction, and it is having a different properties in the axial directions. And in the axial directions only, these carbon nanotubes are very strong compared to that radial directions. Catalytic properties. So by means of using different ligands, we can able to change the catalyst activity of that nanoparticles, which are depends upon the size and properties of that nanoparticles. Sterically stabilizing nanoparticles by using a oligomers or polymers, we can able to prevent the interaction of nanoparticles between each other. And by means of using biometallic nanoparticles, we can able to produce a synergistic effect on the catalysis between the 2 metals. And coming to that, magnetic properties. Super paramagnetisms. So it is nothing but it is a one form of magnetisms which will appear on these small ferromagnetics or ferromagnetic nanoparticles. and this material, that is a nanomaterial, is free from coercivity and remanence. So what is coercivity? It is nothing but a magnetic field necessary to demagnetize the fully magnetized materials. So how much magnetic field is necessary to demagnetize the fully magnetized materials? So that will need to have some resistance. So that resistance we will call it as a coercivity. Remanence is otherwise called as remanent magnetization, or we will call it as a residual magnetism. The name itself, it is the residual magnetism. So that means something will be left. So in the external magnetic field, when we are removing from any materials, so some magnetic materials, ferromagnetic materials, that magnetization will be left in that particular things. So that particular one we will call it as a residual magnetism, and we will call that one commonly as a remanence. So what is the special properties of these super paramagnetic particles means? It will be loosed when that particles are kept at a distance. A high energy of anisotropic particles are combined with these super paramagnetic particles, which is the important class of these permanent magnetic materials that will be produced. And these magnetic nanoparticles will be used in refrigerations, bioprocessing, and high standard density magnetic memory media. So these are all the various areas where these magnetic properties of these nanoparticles are useful. Coming to that optical properties. Refractive index. So these refractive index of the polymers can be tuned by means of distributing a non-agglomerated nanoparticles. So that materials with an non-linear optical properties can be produced by this particular process. And in case if you have taken a gold or a cadmium, selenium nanoparticles in a glass tubes, it may lead to produce a red or orange color. So that is due to the optical properties of these gold or cadmium, selenium nanoparticles only. And similarly, fluorescence, which was exhibited by some oxide polymers of nanocomposites, are semiconducting nanoparticles by a blue shift with a decreasing particle size. So that is also again due to the optical properties of that nanomaterials. And there are some two different effects which are responsible for the behavior of these nanoparticles. What are all those two effects means? One is called as a surface effects. The another one we will call it as a quantum effects. Surface effects, it is nothing but a fraction of the atoms at the surface will cause us a smooth property scaling. And quantum effects, which will showing a discontinuous behavior due to the confinement of the quantum effects in the materials because of that delocalized electrons. These surface effects and quantum effects will affect the chemical reactivity of that materials, as well as that whatever the properties we get discussed. There is a mechanical, optical, electrical, and magnetic properties of your nanomaterials. And now this particular table will give a brief idea about how these nanoparticles are differ from that bulk materials. So first thing is on a melting point. The melting point of a nanomaterial is lower or decreased. Why that nanoparticles melting point is decreased means it is due to the decreased lattice constant. So in the nanoparticles, the lattice constant is decreased because of the decreased lattice constants that have on a lower melting point. Whereas if the same thing if you taken into bulk materials, it have on a higher melting point. Why means here the lattice constant is increased. Mechanical strength, if you are comparing nanoparticles, as I told, carbon nanotubes is the hardest materials in the nature, that it is the ultra hard. Why means it is due to the decreased probability of that defects. Whereas the bulk materials are having usually 1 to 2 times lesser strength, mechanical strength than that nanoparticles. Both nanoparticles as well as bulk materials will exhibit the optical properties, and particularly in the nanoparticles, the color changes will absorb in the spectra due to the surface plasmons resonances. That decrease the electrical conductivity is observed in that nanoparticles. Why? Means that we know that nanoparticles having a larger surface area, and because of that larger surface area, the surface scattering is increased. Because of that increased surface scattering, the electrical conductivity will decrease. Whereas in the bulk materials, ultimately the surface area is less, hence the electrical conductivity is increased. And ferromagnetism is disappear in the nanoparticles due to that super paramagnetisms, whereas it will be appear in the bulk materials because there is no super paramagnetisms in the bulk materials. And if you are looking at thermodynamic properties, the self-purification is possible in that nanoparticles, whereas it is not possible in the bulk materials. And nanoparticles are chemically stable, more stable because of that increased perfections, whereas bulk material is less stable. And surface area is large. A simple example I will tell you that 0.3 microgram mass of that 16 nanometer carbon is 11.3 millimeter square. The same 0.3 microgram of that carbon at a 16 micrometer diameter is only on a 0.03 millimeter squares only. Then you think here it is 11.3, here it is 0.0. So that much surface area is getting in that nanoparticles. And the number of particles which are present in the unit mass, if you are looking nanoparticles, the number of particles present will be high, whereas in the bulk materials it is on a low. How these nanostructure materials are broadly classified? These nanostructure materials are broadly classified into one of four major types. That is a nanoclusters, nanorods, or it is a nanotubes, and it may be a nanowires. What is that nanoclusters? The name itself, it is the clusters. It is the smaller nanoparticles, and these nanoclusters are resemble those of molecules. These nanoclusters are bridging a gap between the nanoparticle and the atoms. So the gap between the atoms and nanoparticles are filled by these nanoclusters only. And in a simple way, if you want to tell, means we can tell that nanocluster as a intermediate state of matter between the solids and their molecules, which are constituted by these nanoclusters. And the size of this nanocluster is sub-nanometer. That means below that nanometer. So for example, 0.1 nanometer, 0.01 nanometer to that 10 nanometer. In the diameter, the nanoclusters will be in that sub-nanometer to that 10 nanometers. These nanoclusters are useful in many fields, particularly in the optoelectronics, catalysis, and material sciences. And this is the picture of gold strontium nanoclusters. Nanorods, that is nanomaterials or nanoparticles which are present in the rod sheet. So the size is usually between 1 to 100 nanometer. In case of a nanocluster, it is sub-nanometer to 10, but here it is 1 to 100 nanometer. That's why we told that nanocluster is the smallest of that nanoparticles. And semiconducting materials are metals. From that only these nanorods are synthesized. Or we can able to synthesize these nanorod by means of a direct chemical synthesis also. The length and width ratio for this nanorod is maybe an 1:3 or 1:5. So that is the size of this nanorod. So between that length and the width, maybe on a 1:3 and 1:5. So the length is 3 and the width is on a 1. Similarly, the length may be on a 5 and the width is on a 1. And these nanorods are mainly useful in the cancer therapeutics. In the application part, we can discuss this elaborately. And the major application of these nanorods are in the display technology, such as what the things we are using now in the mobile phones and the LED screen, LCD screen. In all these cases, these nanorods will play a major role. And this is the diagrammatic representation of a gold nanorods. So at that 50 nanometer scale, that the gold nanoparticles are present in these spherical heads. At 200 nanometers, it is like a backbone-like structures, and it's a dumbbell shape in that again at 50 nanometers, and in 200 nanometers that may be on a cuboidal shapes. And these four pictures represent the that is the various stages of the anisotropic oxidation of a gold nanorods. These are all the various stages of the transfer overgrowth of these gold nanorods. Nanotubes. So generally nanotubes commonly we will call it as a carbon nanotubes only. It is otherwise called as bulky tubes also. And these carbon nanotubes are synthesized from the fullerene, which is having a similar structure like that of geodesic domes. Hexagonal graphite molecules are attached at the edges, which are present in miniature cylindrical carbon structure, and that only produce a carbon nanotubes. And how these carbon nanotubes are produced means by means of rolling up of the sheets of a graphene, which will form the hollow strands with only one atom thickness of the wall, but it may look like a powder or a black soot. So this is the picture of that in nanotubes. So that already we have discussed. So by means of rolling up this graphene sheet, we can able to get that carbon nanotubes. And here you look, there is a multi-layers. So here there is a 3 layer of that carbon nanotubes is there. So this one we will call it as a multi-layer nanotubes. Nanowires. So here the ratio between that length and thickness between is the 1:10. So that's why we are calling as tens of nanometers or less. And again, the length is less than that 1,000 times. So the wires, the length will be, for example, it is on 1,000 nanometers, but the width will be only on 1 nanometer. So that much things will happen in that nanowire between the ratio of your length and width. These nanowires are further subdivided into 3 major types as metallic nanowires, semiconducting nanowires, and insulating nanowires. And gold, platinum, and nickel nanowires are example for metallic nanowires. Gallium nanowires and silicon nanowires are example for semiconducting nanowires. Titanium dioxide nanowires and silicon dioxide nanowires are example for insulating nanowires. When these molecular units are repeating, that molecular units may be an organic or it may be an inorganics that may produce a molecular nanowires. So when these nanowires are repeatedly present, from that we can able to get that molecular nanowires also. An example for that organic is on a DNA and inorganic is that molybdenum 6s9xls. This is the diagrams of silicon nanowires, and this one is on a neuron reading nanowires that will accelerate the development of your drugs. Coming to that our part that is on a nanochemistry, how these nanomaterials are synthesized, what are all the different methods which are available for this synthesis of your nanomaterials. There are mainly two approaches are there. One is called as a top-down approach. The another one we will call it as a bottom-up approach. The name itself it is the top-down. So from the top it was coming to on a down. So top is on a bigger and down we can consider as on a small. So a larger or externally controlled one. So from that a big molecules it was assembled to create a nanoscale devices. So larger particles are broken into get that a smaller nano range. So that is called as top-down approach. What is that bottom approach? It is reverse of that top-down approach. From a nanoscale materials, we are assembling in a such a way that things to get on a big molecules. Okay, so that is called as a bottom-up approach. And here that nanomaterials are assembled in a complex manners. So that we will elaborately discuss. This diagrammatic representation will easily useful for you to understand what is the top-down approach and what is bottom-down approach. So you look in the top-down approach, I told you bulk materials are present. And initially that bulk materials were converted into a powder. How? By means of transforming some energy. then that powder will be exploited into a multi-layer or a mono-layer. How? Again, transforming into a smaller fragments, that powder we are transforming. So from that, a nanomaterials can be synthesized. So from a bulk, which are big, a smaller particles we are synthesizing. So from your top, we synthesized the downward. So that's why we are calling this approach as a top-down approach. The reverse one you can think of, bottom-up approach. So bottom, smaller things. So the precursor which are containing molecules, that is an atom level we are taking. So that atoms or molecules we are ionizing using an energy. From that the ions, radicals or electrons can be formed, and by means of condensing the formed one as a clusters we can able to get, and from that clusters and nanoparticles are synthesized. So these are smaller than these nanoparticles. From that nanoparticles are synthesized. So that's why we are calling this one as a bottom up approach. And some examples of these nanoparticles are graphene nanosheets, diamond and fluorine, all those things. So this is another way of representing a top-down approach and a bottom-up approach. You look here, the bulk materials which are converted into fragments. From the fragments, nanoscale structures are synthesized. And in case of your bottom approach, you look, the atoms are there. From the atoms, we are getting clusters. From the clusters, we are able to prepare a nanoscale structures. And this is another representation how that top-down and bottom approach we can easy to understand. So bottom-up approach, you look, atoms or molecules are present. By means of ionization, utilizing some energy, it was converted into clusters. And these are the various techniques which are useful for that bottom approaches. Vapor deposition, solvent process, aerosol process. We can able to synthesize a different shaped nanoparticles. And in case of a top-down approach, a bulk materials, by means of transforming energy, we are converting that into a powder by using the techniques such as sputtering, chemical etching, and ball milling. Then we are able to get a different shaped nanoparticles. Let me will discuss this top-down approach and a bottom-up approach elaborately. So top-down approach. So this top-down approach will utilize microfabrications or the traditional workshops. For what purpose? In order to get what the shape we are deciding, what the size we want to get it. So traditional workshop means it is nothing but cutting or milling or shaping that particular materials. So by means of that one, if you are getting a nanoparticles from the bulk materials, that one is called as a top-down approach. A simple example, if you want to tell, means for the top-down approach, inkjet printing we can tell. That is a very simple example for a top-down approach. Photolithography is also a technique which we got it from this using this top-down approach. And in the top-down approach, there are three important methods is there. Labor ablation method, chemical vapor deposition method, and the electro deposition methods. Let me discuss elaborately about these three methods. Laser ablation method. The name itself it is there, laser ablation. That means in this method we are using laser irradiations. So we know what will happen when that laser material is irradiated with a laser. Ultimately, it will produce heat. Because of that heat, the materials will be evaporate. When it is evaporating, we can able to get that smaller particles. So this laser ablation method is mainly useful for solid particles only. Sometimes we can use these things for liquid samples also. So that is the thing. When that materials is irradiated with a beam of lasers, that will remove materials from the samples. Why it is removed means because the heat will be developed in that materials. Because of that heat, it will be evaporated. When we are sublimed that one, we can able to get that smaller particles. So this is about the diagrammatic representation of that labor ablation method. So here if you are looking, this one is on a laser beam, and here that argon gas was filled, and this is called as a furnace, and here we are keeping that material. So here we are keeping a graphite targets. So when that laser beam is placed on that graphite targets, it will produce a heat. Because of that heat, it will evaporate some of the materials, and condensing that particular materials, we can able to develop nanotubes, and that nanotubes we can able to collect. So this is solid. I told the laser ablation method sometimes may be useful for a liquid also. How it will be work out in the liquids? In the liquid, if we are placing a metal plate and we are stirring that liquids. So from the top, we are passing that laser to the materials, and ultimately it will generate a heat and it will produce that nanoparticles. We can able to control all these things by means of this controller. The next method is chemical vapor deposition method. Simply we will call it as CVD methods. So this particular method is generally used to get a high purity solid materials. When we need a high purity solid materials, or the other way we can tell it as a high performance solid materials, this is the one of the preferred methods. So here, these particular methods sometimes may be used to produce a thin film also. So what happened means, again in the name itself, it is chemical vapor deposition. So here we are using a chemical vapors for producing that nanomaterials and nanoparticles. Okay, so one or more volatile precursors are reacted, or it was be decomposed on the surface of the substrate that ultimately produce whatever we need. And this particular method is useful for producing a synthetic diamond and microfabrication process. So these two things are mainly by using this CVD method. So here that I told you, the vapors. Okay, these are all the vapors which we are going to use, and this we can consider as an initiator, and you consider this one as an monomers. So using these vapors, we are passing through the vacuum chamber. So from that we can able to get that what the things we are looking at nanomaterials. So here this is another way. So here this was be enlarged, that one. Here it will be on a cooled storage, that is 50 degrees Celsius, whereas here the filaments are at the temperature of 200 to 300 degrees Celsius. So when the vapors are passing, that the materials will get, and finally after cooling we can able to get that nanoparticles. Electrode deposition method. So here we are using the electrical current for the deposition of nanoparticles. So generally which will be useful for coating on any objects. And what purpose it is useful? So if you are coating that materials with these nanoparticles, what will happen means it may prevent the rusting and corrosions. Okay, so as we people may aware of that, the electrically charged particles will move according to the what's that, whether it is moving towards on a cathode or electrode, the same principle will be applicable here. So that the ions are moving and it will be deposited on that materials. And it is otherwise called as an electrophoretic deposition also. And then till now we discussed about top-down approach. So from a big compounds or from a bulk materials, we synthesize nanoparticles. Now we are going to discuss about a bottom-up approach. So molecular recognition or molecular self-assembly concept will be utilized in this bottom-up approach. And in this approach, a chemical properties of a single molecules only will use. Using that single properties, the chemical properties of that single molecules, we are assembling that single molecules into an organized manner to get some useful conformations. And it will be rely only on the positions. This comparing top-down and bottom-down approach, this bottom-up approach only produce the devices in a parallel manner, and it is much cheaper. If any method is having an advantages, ultimately it may also having a disadvantages, because it may give a cheaper and it will produce the devices in a parallel manner, but it is a complex one and will depends upon that size of the desired assembly also. And there are two different methods in the bottom-up approach. One is on a precipitation method, the other one is on a thermolysis method. And there are two different methods of a thermolysis. One is hydrothermal method, the other one is on a salvothermal methods. Precipitation, name itself it is that nanoparticles are precipitated. Okay, so generally this method will be useful utilizing many reducing agents for reduce the metal salts. Okay, and a very good example here I was quoted is that 0.1 mole of a cobalt nitrate, when it was treated with a 0.2 mole of a ferric nitrate, we are adding an sodium hydroxide in order to get that corresponding pH of an 12. If we are stirred vigorously at an 80 degrees Celsius, we can able to get an cobalt ferrite nanoparticles after a 3 hours. So here the nanoparticles are precipitated. That's why we are calling that one as a precipitation method. Thermolysis method. Name itself is the thermolysis. Lysis means breakdown by means of that thermal heat. Okay, so high temperature production of a nanoparticles from the solids or liquids which are having a molecular anions or metal cations or metal organic compounds. That decomposition is the process. So that one only we are calling as a thermolysis. A very good simple example is decomposition of a lithium azide by using converter into a lithium. Hydrothermal and salvothermal. There are two types. Hydrothermal, hydro, that it is nothing but water. So here in this technique, we are using an aqueous solutions. Under high pressure, the single crystals are synthesized, which are depends upon the solubility of the minerals present in the hot water. So cellulose fibers was dispersed in the water and ethanol. To that, we are adding titanium isopropoxide in ethanol, and we are stirring that one for 1 hour. And after 1 hour, we are transforming into that autoclave. Then in that autoclave, we are keeping it for 80 degrees Celsius for 24 hours. Then we are taking and we are washing with that ethanol. We will get a titanium dioxide immobilized cellulose fibers. And that one we can use it for making a sheet-like models. Salvothermal. The difference between hydro things and here salvo is there we are using an aqueous solution. Here we are using an non-aqueous solution. That is the only difference. But this salvothermal method having more control, precise control over the distribution of a shape, size, and the crystallinity of a nanostructures or a nanoparticles. By means of changing the experimental parameters such as reaction time, reaction temperature, precursor type, surfactant type, solvent type, these properties can be modified. Nanostructured carbon, graphene, titanium dioxide, other materials all are synthesized by using this salvothermal method. So a simple example I was explaining that salvothermal method. 99% zinc nitrate was treated with sodium hydroxide, which one we are stirring for 30 minutes. We will get adding surfactants. Then we are sonicating for 45 minutes. We will get a linear Teflons. After that one, when we are transferring to a salvothermal reactor and we are doing that reaction for 180 degrees Celsius for 3 hours, and after 3 hours we are washing the samples with water and ethanol, and we are drying at 60 degrees Celsius for 24 hours in a dry oven, we will get a zinc oxide nanoparticles. After getting the nanoparticles, we are categorizing that nanoparticles and we are using for different applications. And any technique if we taken, it may have some disadvantages. Like that, these nanomaterials may also having some disadvantages. What are all those things? The first important one is instability of the particles. Why these nanomaterials are unstable? Because of the kinetics which are associated with that nanomaterials. The kinetics are rapid, that's why it is unstable. Nanomaterials are explosive. Why it is explosive means because they have a higher surface area. Because of the higher surface area, they can have a more contact with that oxygen, and there is a chances of that exothermic composition. These nanomaterials that impurity is the another disadvantages. Why it is that nanoparticles or nanomaterial is impure means because it can easily highly reactive. It can easily react with many impurities. And nanomaterials are biologically harmful. Yes, definitely, because it was transparent to the cell dermis and it is having a high surface area. Because of that, it may have a surface activity is increased. And nanomaterials, there is a difficulty in synthesis, isolation, and applications. And again, recycling and disposal is also till now there are no specific method is there. That's why it is on another disadvantages of these nanomaterials. And application of the nanoparticles. So nanoparticles I already told you that it is having no specific field. It will be having an application in all field. It have an application in physics, have an application in technology field, it have an application in electronics, and it have an application in the medical field. Like that, it have so many applications, these nanoparticles. So in the chemistry, materials and environmental testing, it have applications, and in the automobiles and machineries also it having an applications, because the power saving, everything will be increased when that particle size are less. And it is having an electronics and communication, as I already told, nanorods, which having a major application in that displays things. And in pharmaceutical field, these nanoparticles are useful in the treatment of cancers particularly, and in the energy field also it have so many applications. And these are other diagrammatic representation which will discuss about the application of nanoparticles. And I was not going in depth about each and everything, and I was fortunate to go about only in the medicine field. Okay, so here it may have an application in the environment as a catalytic activity. It will useful for removing toxic pollutants, and in agriculture, these nanoparticles are useful for gene delivery and labeling of your cells and biomolecule detections. And in the consumer goods and electronics, it will be useful in producing cosmetics and fast-moving consumer goods and photonic crystals, and in that nanomedicines. That is the most important thing that I'm going to concentrate these things only: targeted drug delivery, gene therapy, cancer therapy, antioxidant activity, antimicrobial activity, mosquitocidal uses. And in case of your veterinary medicines, the polymeric nanoparticles are useful, liposomal nanoparticles are there in the veterinary medicines, and micellar nanoparticles and metallic nanoparticles such as iron oxide, zinc oxide, silver nanoparticles and gold nanoparticles are useful, and liposome nanoparticles and dendrimer nanoparticles also is useful in that veterinary medicines and in the medicines and biology. So as I told that it will be useful for the treatment of a cancer, and it will be useful in the drug delivery system, particularly a targeted drug delivery systems and detection of your biomolecules, and it will be useful for molecular imaging and many more. And it is also having usefulness in the regenerative medicines, and these nanoparticles will be useful for the diagnosis of a disease, and it will be also useful for the antimicrobial resistance. So that is a major problem what we are facing in our day-to-day life, that is a microbial resistance. This nanoparticle will be useful for fighting about those things, and it is also useful for the gene delivery also. So I think with these case studies, I will complete my presentations. So till now we discussed about that what is that nanochemistry, nanoparticles, nanoscience, nanotechnology, and what we used these nanotechnologies in the laboratory and what we produced with that things. I will explain with practical examples. So this is the latest work what we get done in our laboratory, that we had designed by means of an in silico modeling, and we utilized a functionality theory, folate receptor targeted myricetin loaded bovine serum albumin nanoparticle. We formulated this myricetin loaded bovine serum albumin nanoparticles for a cancer treatment. Okay, and this is a latest one, and it was going to be published in the Journal of Nanotechnology, which is having an impact factor around 5, which is published by an IOP Publishers. And along with my research collaborators, we get done this particular work, and here in this work we get done so many things, and I'm going to explain about that nanoparticles part here. So I will give a brief introduction about what we get done. So targeted drug delivery system, which is a very promising field in the research, because here we are particularly targeting one particular system for delivering drug molecules. And we know about just now that nano-engineered materials having a remarkable potential for the treatment of various malignancies. So these two things are already proved. So what we done means we are planned to synthesize these folic acid conjugated bovine serum albumin nanoparticles, and we are using these nanoparticles to encapsulate myricetin. This myricetin is nothing but polyhydroxy compounds which was available obtained from the plant, which is having an anti-cancer activities. So that we are encompassing these particular molecules by using that nanoparticles. So after that, what we are done means we are exposing these particular nanoparticles to a overexpressed folate receptors in the breast cancer cells. And we prepared that nanoparticles by means of using a modified desolvation cross-linking technique. So that is the technique what we use for synthesizing these nanoparticles. And folic acid is conjugated with that N-hydroxysuccinamide, and after conjugating with that N-hydroxysuccinamide, we confirmed the structural characteristics by using a various techniques such as UV, IR, NMR techniques. We confirmed the structure of that folic acid conjugated N-hydroxysuccinamide. Then we are checked that biocompatibility of these folic acid conjugated N-hydroxysuccinamide with that myristoyl loaded bovine serum albumin. Then we have formulated that folic acid conjugated myristoyl loaded bovine serum albumin nanoparticles. We studied that morphology, size, shape, physiological stability, and drug release kinetics of that particular things. Then we used a molecular docking studies also to know about the effectiveness in the folate receptors. And from the study, we find out that these particular nanoparticles fastly released the myristin in an acid medium. Ultimately, in the physiological medium also, it is going to release that myristin in the faster manner, and it will decrease the viability of that MGF cells after 24 hours, and it increased the uptake of that myristin molecules. And after incubation period, we also noted that apoptotic morphology of the condensed nucleic and distorted membrane bodies is also observed. And in addition, it is also increased the reactive oxygen species release, and which will contribute the loss of mitochondrial membrane integrity. So finally, we concluded that this particular myristin, it increases the anti-cancer activity of that. The drug molecule what we get taken is myristin. So this is the schematic representation of the nanoparticles what we get synthesized. Folic acid initially, it's on 3 steps. The first step, folic acid was reacted with an N-hydroxysuccinamide in presence of a N, dicyclohexyl carbodiamides. So we got on a folic acid with an N-hydroxysuccinamide, that is folic acid N-hydroxysuccinamide. That is the first step. In the second step, myristin was reacted with an bovine serum albumin, and we got an myristin loaded bovine serum albumin nanoparticles. Then these nanoparticles have an amino group. That amino group was reacted with that folic acid N-hydroxysuccinamide, where that will replaces that N-hydroxysuccinamide, and these nanoparticles, myristin loaded nanoparticles will attach. And that one, when we are administering to the cells, it will react with that folate receptors, and it will easily enter into the cells, where it will release that nanoparticles, and from that nanoparticles, that myristin will be easily react with that materials which are present in the nucleus and the mitochondria. Thereby, the cancer activity of the myristin was increased. So how that nanoparticles was synthesized? So initially, what we done means 1 mg of the myristin we taken, to that we added on a 5 ml of the ethanol, and we are doing an sonications for dissolving purposes. Once the myristin was dissolved in an ethanol, then we added a 50 ml of a deionized water, which will contain that 0.25 gram of your bovine serum albumin and 0.1 ml of your glutaraldehyde, and we added these things in a drop-wise manner. And we are stirring, and we incubated this particular mixer for 12 hours with the continuous stirring in order to get a clear solutions. So what happened means during these 12 hours that a slow encapsulation will take place for that myristin, and ultimately we may able to get that hydrophobic drug myristin which be slowly encapsulated with this ethanol and all those things, and we will get nanoparticles. So to remove that non-encapsulated myristin, that we are centrifuging that things at 1500 RPM, and we washed with an deionized water for 3 times. And whatever the myristin bovine serum albumin nanoparticles we got, we are dispersed in 3 ml purified water for further use. So that is the step 1. Step 2, that we have to prepare that folic acid conjugation. So for that, 1.5 gram of the folic acid be taken, and we dissolve that 1.5 gram of the folic acid in a mixture of 25 ml of dimethyl sulfoxide, 0.45 gram of N-hydroxysuccinamide, 1.05 gram of dicyclohexyl carbodiamide, and 0.25 ml of triethylamide. After mixing that one, we activated this solution. How? By means of placing in a water bath at 40 degrees Celsius, and we continuously sit for 48 hours at 80 RPM. And the byproduct which will be formed is dicyclohexyl urea that was removed by means of a filtration. Then the resultant filtrate will contain folic acid and hydroxysuccinamide solutions that will be lyophilized for 48 hours, and it will be stored in the dark place. Then whatever we got in step 1, whatever we got in step 2, that is step 1 we got a myristin encapsulator bovine serum albumin nanoparticles, and step 2 we got the folic acid and hydroxysuccinamide solutions. So here we are going to mix both the things in order to get what we gain. So 5 mg of that myristin bovine serum albumin nanoparticles we get taken. We are mixed or dissolved in that buffer. We can use either a carbonate buffer or a bicarbonate buffer. The pH should be 10. To that, we added a DMSO solution of that folic acid and hydroxysuccinamide in the concentration of 1 mg per ml. And we stirred for 2 to 3 hours at room temperature. And DMSO was removed by means of a dialysis. And we used the Cefadex G50 column to separate that unreacted folic acid and hydroxysuccinamide. And finally, we will get that folic acid conjugated myristin bovine serum albumin nanoparticles, which are centrifuged for 30 minutes at 2000 RPM, and finally it was redispersed in purified waters. So these are all the other scientific works what we had done, and it was published in good journals based on these nanoparticles. So this was in the year of 2020, which was published in 3 Biotech. There we formulated and characterized the folate receptor targeted pegylated liposomes encapsulating bioactive compounds from Capacis albergili for cancer therapy. The another one work we had designed, synthesized in silico modeling, functionality theory of a novel folate receptor targeted routine encapsulated folic acid conjugated keratin nanoparticles for a cancer treatment. And we have also done and work on graph theoretical analysis, in silico modeling, and formulation of pyrimidine nanoparticle as a P38 alpha MAP kinase inhibitors. It is a quantitative proteomics approach by means of a well-known TIMI publishers. And design and in silico modeling of indoloquinoxaline incorporated keratin nanoparticles for modulation of a glucose metabolism in 3T3 L1 adipocytes, which was published in Biotechnology Progress in that year 2019. And we also formulated and evaluated oriconosol ophthalmic solid lipid nanoparticles in cytogen. So these are all the various work which we get done in this nanoparticles and nanomaterials range. And from the things, finally I was conclude that nanotechnology has the potential to revolutionize a diverse range of field, which was ranging from a healthcare to the manufacturing. But the safety of a nanomaterials and nanotechnology is still being debated, tested, and assessed. So my presentation will not be complete without telling or without acknowledging some people who are always with me and who are encouraging me. And in that regards, I want to tell my thanks to our chairman, Sir Sri M.N. Raju Garu, MNR Educational Trust, and Sri Ravi Varma Mantanaharu, Vice Chairman, MNR Educational Trust, and my principal, Dr. V. Alagarsamy, who is always back to me and support me for each and every my growth, and he's on a role model for me, and he's on my group. And in addition to that, I also thankful to my research collaborators, Dr. T. Panneerselvam, who is on a principal of Saraswati College of Pharmacy, Hairpur, Delhi. Dr. K. Selvaraj, who is on a research scientist, in that Kalasaringam Academy of Research and Education, Krishnan Goyal, and Dr. P. Parasuraman, who is an associate professor in MS Ramaya University of Applied Science and Bangalore. And last but not least, I am also thankful to my family members, particularly my wife and my children, those who are back to me and who are always supporting me for my all activities, and they are sacrificed their many times spending on a valuable time. So I'm also thankful to my family members also. So with these things, I will complete my presentations, and these are all the various references from where I was took that materials. And now I was handed over the sessions to the organizers for a question and answer sessions.
Yes, thank you, Saravanan, for your valuable contribution in this topic, and the effort what you had taken for the preparation for this topic is amazing one. And you are simply inputting the matter of nano, but you had discussed in and out about that particular topic in detail as a macro level. So thank you, friend, for sharing such a knowledge in this platform. Definitely, it could be an amazing presentation. I assure that it could be beneficial to our delegates in a great way. Thank you, Saravanan, for your great contribution to this. And now it's the time for a questionnaire session. The suggestions and the questions from our participants are dumped into our question answer box. So I invite Mrs. Gladys Kalpana, Professor, Department of Pharmacology, Nandha College of Pharmacy, to join with us for this discussion. Please, ma'am.
Yeah, good morning, sir. Good morning, everyone. Good morning, all the participants and the panelists. And I'll throw one by one questions. And this question is from Professor Dr. V. Ravichandran from AIMS University, Malaysia. Yes, asked you two questions. The first one is, what is your suggestion on how to reduce the toxicity of particularly metal or inorganic nanoparticles like silver, gold in medical applications?
I'm not getting madam. Voice was break.
Yeah,
Can you please repeat the questions?
Yeah, what is your suggestion on how to reduce the toxicity of particularly metal or inorganic nanoparticles like silver or gold in medical applications?
Yes, if you want to talk about on a toxicities, that is the thing what I was discussed in my presentation itself. That everything it was we are working on that toxicity levels only. So when we want to reduce that, what is that surface areas, particle size, and what the mediums we are using for preparing that silver and was that gold nanoparticles. So the by means of selecting that particular pathways, so what we are using in that way, and many scientists that nowadays that it was working on the toxicities only. So because of that large exposed surface areas, so that is what is the advantages of the nanoparticles as well as the disadvantages of the nanoparticles. So in that way, by means of controlling that surface areas and other properties which are associated with that silver and gold nanoparticles, we can be able to reduce the toxicities.
Okay, thank you, sir. And the second question is from the same professor, Dr. V. Ravichandran, regarding the metabolism of metal nanoparticles, how to improve the metabolism of metal nanoparticles in human body?
So, frankly, if we want to tell, means that I was not that much aware of that metabolism of these particular nanoparticles. But generally, what will happen means because of that nano size, when it was entered into our biological systems, that will be easily metabolized and it will be removed from our biological systems. I think hope I may not aware of that particular things in that metabolisms. And in case if you are forwarding his email ID, then I will refer and I will send the details.
Okay, that he might get the answers from your side, sir. And the next question from our participant is, except in cancer therapy, is there any other application of nanoparticles in pharmaceutical sciences?
Yes, we have on many field. It is not only for on cancers only. Any targeted drug delivery systems. So in whatever the things. Okay, but the major uses we are using these nanoparticles for on cancer therapies only. Any targeted drug delivery systems we can use these nanoparticles.
Thank you, sir. And the next question from our participants is how to study medicinal herbs with nanochemistry?
So medicinal herbs again that for an all active compounds that the basics is on a natural products only. Okay, so first we want to know about we have to extract what which compound is responsible for that natural products. Whatever the case studies I was explained that myricetin, it is also on a natural products only. What the works we get done, all those are on a natural products only. So in that natural products only, we took that myricetin we taken, and in that myricetin we can encapsulate it with things, and we prepared that nanoparticles of that myricetin, and we targeted that myricetin to on a cancer cells, and thereby that myricetin is having anti-cancer activities because of the targeted delivery systems, and because of these nanoparticles that the effectiveness of this myricetin was increased. So in the same way, any natural products we can take, and we can able to produce on a nanoparticles of that particular natural products, and which way we want to target that particular tissues, we can target that particular tissues, and we can achieve our effects.
Yeah, thank you, sir. And the next question from the participants is nanoparticles in parenteral preparation can exist any elements?
So in the parenteral preparation, if you are taken that nanoparticles, element can exist means, yes, it may exist. And again, the thing is in which way we are preparing that nanoparticles and what the process we are using to synthesize that nanoparticles. So that only will play a major.
Yeah, okay, sir. And the next question is having many applications and users, why nanotechnology has not yet reached wide? And what is the use of nanotechnology in agriculture?
So for these questions, I already discussed that answer in that my presentation itself. So because of the disadvantages, what I was discussed about that in nanomaterials, because of the disadvantages, and there is no specific way for safe disposal of these nanomaterials, and we can't tell that it was not distributed widely, because nowadays we are using what's the mobile phones and the screens, LED screen, all those things, because it was prepared by using these nanotechnologies only. And in case of agriculture also, I told that there is gene technologies and all these things which was applicable by using these nanomaterials in the agriculture. So that will also increase the crop field, all those things. Because why, when we are using this things in an agriculture, because of the increased surface areas, the effectiveness will increase, and it may result in a good field.
Yeah, thank you, sir. And the next question from our participants is there any specialization in nano or polymer chemistry after B Farm?
After B Farm, yes, I think in India, I was not aware of that. I think in the abroad, we have a special course on this nanotechnologies itself. It is that there is a master degree is that. For that one, we must be having some chemistry backgrounds. I don't know exactly whether B people, pharmacy people are eligible for that particular. So if what is that? If you are sending that particular persons, I will search in that internet, and I can able to send the details. Which university is offering this particular courses, and what are all the eligibility criteria?
Yeah, thank you, sir. And the next question from our participants is anti-cancer drug can be prepared as nanoparticles or nanorods or nanotubes.
So these rods and tubes, we can't give it to the anti-cancer therapies. Generally, we will go with nanoparticles only. That the things why means these nano rods which have uses in other fields, and when we taken these things as what is nano rods or nanotubes,
what is that? Even we can, there is no need of only nanoparticles. So that I think, hope if you understand the basic difference between these nanorods, nanotubes, and nanoparticles. Nanoparticles having in the 3 dimension itself having a nanoscale. That means the length, diameter, width, and height. All these 3 dimensions are in the nanoscale range. Means we are calling that one as a nanoparticles. So what will happen when all these trees are in the nano range? Ultimately, what will happen? The surface area will be more. In case if you take a nanotubes, the length will be higher. It is not in the nano range, but only in the width is in the nanoscale. Okay, so because of that, the surface area of the nanoparticle compared to that nanotubes is more. So hence, if you prepare as a nanoparticles, the effectiveness will be more compared to that nanotubes and nanomoles.
Okay, thank you, sir. And this is the last question from our participants. They are asking about advantages of nanoparticles over intravenous preparations.
Sorry.
Yeah,
Madam, can you repeat the questions?
Advantages of nanoparticles over intravenous preparation, IV.
So again, that surface areas only, because that nano level, if you are talking, means without talking about on a surface area, it will never comes. Okay, as when that nanoparticles, when we are administering the things inside, so that having a more surface area, ultimately it will be exposed easily, and the effectiveness which was produced by that nanoparticles, we can able to get on a good effects. And in case of IV, we can't be able to get on a targeted drug delivery things. Whereas by using these nanoparticles, we can be able to target that drug delivery system. So that is the major advantages of these nanoparticles.
Okay, thank you, sir. And one more question from our participant. When it is necessary, one sec. When it is necessary to go for TEM for nanoparticle study.
When usually we people work can go ahead with what is that SEM, scanning electron microscopy studies only, we will go ahead. When we want to study about on a what is that this size, shape, everything in an what's that when if we are not getting enough good results in that same technologies, and if we want to further know about these nanoparticles, in such cases we have to go ahead with on a chemistries instead of a SEM. So if you are getting everything in that same itself, it was clear there is no need. And in case if you are not getting anything, or if you are facing any problems in that same technique, in such cases we can go ahead with that.
Thank you so much, sir. Thank you so much for your patience for answering all the questions of our attendees.
Yes,
Thank you, ma'am, and thank you, Saravanan, for your valuable clarification. Now this is the time for gratitude. Now we invite Dr. Jagadishwaran, Professor, Department of Pharmaceutical Analysis, Nandha College of Pharmacy, to deliver the vote of thanks. Please, sir.
Yeah, warm good afternoon to all. I deem it a great honor and privilege to propose the vote of thanks. We would like to thank our management, Sri Nandha Educational Trust, for providing the facilities to organize this national webinar. Thank you, Dr. G. Saravanan, for accepting our invitation in his busy schedule and providing the valuable information about nanochemistry. Once again, thank you, sir. I am extremely thankful to our principal, Dr. T. Shivakumar, for his enormous support and guidance. Thank you, sir. Our special gratitude to our Chief Administrative Officer, Dr. K. Krishnamurthy, for giving valuable guidance and cooperation. Thank you, sir. Our sincere gratitude to the participants in and around the country. Last but not least, I would like to thank our organizing committee members for their technical support and moral support. Thank you, one and all.
Thank you, sir. Thank you for your kind gratitude to each and every one. Thank you so much, sir. We are in conclusion of this session. I express my appreciation to all of our delegates for their patient listening and cooperation throughout. For your reference, today's session is present in our YouTube. Go to Nandha TV channel, to watch and kindly like and subscribe our channel for our further updates too. Also, I extend my gratitude to our eminent speaker, Dr.G.SARAVANAN, my friend, for your valuable time with us, for your awesome session also. And I have to extend my gratitude to our beloved principal, Dr. T. Shivakumar, for his valuable encouragement to reach our milestone each and every day. Thank you to our team members for your wonderful cooperation too. So this is a time for bye, and the delegates are so eager to fill their feedback link to receive their e-certificates. They are given by more messages in our chat box too. It will be activate within a seconds. So after the completion of this program. Thank you so much. Bye. Take care. We will meet you in another session shortly. Thank you so much.